Hi guys, so we are studying chapter 2 today, which is chemistry. Before you panic, we're only studying the basics of chemistry, so don't worry. But first we have to define chemistry. Chemistry is just the science that studies composition and properties of substances or matter. Matter is anything that takes up space and has mass. So basically chemistry just studies the properties of that matter and what they're made of. But why do we have to study chemistry? Well, first of all, cells are living things. Cells are the basic structural and functional unit of life. So we are made up of cells. Cells are made up of chemicals, which are non-living. So we have to understand those chemicals that make up the cells that make up our body. Our body also relies on chemical reactions, which remember is our metabolism. Metabolism is just the sum of all of the chemical reactions in our body. Now those chemical reactions help maintain homeostasis, which remember homeostasis is just that happy, steady state. So in a nutshell, we have to study chemistry so that we can understand the chemicals that make up our cells, that make up our body. And we also have to be able to understand the chemical reactions that keep occurring in our body that help maintain homeostasis and help keep us healthy and happy. So, all living things and non-living things are composed of matter. Now, matter is anything that takes up space and has mass, like I said before. However, matter takes on three forms: solid, liquid, and gas. A solid has a definite shape and a definite volume. Liquid has a definite volume, but the shape depends on whatever container it's in. But a gas does not have a definite shape or a definite volume because as it goes into the air, it kind of dissipates. Now, an element is a building block of matter. There are 92 naturally occurring, which just means that they occur in nature, and there are 118 total. We have made the other ones up in a lab. Each one of them has a unique chemical symbol, which is just a letter abbreviation. It's usually one letter, maybe two letters. And there are 26 found in the body. Now, the major elements, which make up 96% of the total, are carbon, hydrogen, oxygen, and nitrogen. There are also lesser elements, which make up about 3.6% of the total: calcium, phosphorus, potassium, sulfur, sodium, chloride, magnesium, and iron. But just because these are lesser elements doesn't mean they're not important. We're still going to be looking at how those impact our body during this whole year. Then we have trace elements, which make up only about 0.4% of the total. But again, even those are in such small amounts, they still have to be monitored. So if you look at the little guy over there on the side, the majority of the element that makes up our body is oxygen, 65%. Then we have carbon at 18%, hydrogen at 10%, nitrogen at 3%, and then all the other elements that are in our body. So this is just a chart showing you the element, its abbreviation, how much percent of body mass it makes up, and what it does, why it's important. Now, the four major elements, oxygen, carbon, hydrogen, and nitrogen, are really going to be our focus in this chapter. However, we're going to be learning about all of these elements and the roles that they play in our bodies whenever we get to those respective chapters during this year. So we're going to learn about calcium and phosphorus and potassium and sulfur, sodium, chloride, and how they impact our body's functioning throughout the year. So an atom is the smallest unit of matter, and this is what different elements are made up of. All atoms of the same element are going to have a similar atomic structure. Now, atoms are made up of three things: protons, neutrons, and electrons. Protons have a positive charge. So if you just remember, proton starts with a P, positive. Neutrons are neutral. They don't have a charge. So neutron, neutral, they both have the same beginning. And then that leaves electrons with a negative charge. Now, protons and neutrons are found inside the nucleus. So that's the big ball in the middle there in those pictures. Now, the first model, the electron cloud model, is the more realistic model. However, we use the electron shell model in our books because it's much easier to comprehend that way. The electron shells are where the electrons are found. So on that picture, the protons are reddish, the neutrons are bluish, and then the electrons are those black dots that are orbiting the nucleus. Now, the electron shells are also called energy levels. They contain orbits, but we don't really care about the orbits so much. That would be for an actual chemistry class. But the electrons fill from the inner shell to the outer shell. Now, what's important to note is that first electron is full, or that first energy shell is full with 2 electrons. All of the rest contain 8. Now, technically, that third shell holds up to 18, but it's full for our purposes with 8 electrons. Now, the outermost shell is also referred to as the valence shell. So that's why it says the third is full with 8 valence electrons. Valence just means the outermost shell. Now, the first shell fills with 2, and then the rest are going to fill with 8. And that's going to be a very important distinction coming up. Now, why I say that first picture, the electron cloud model, is more accurate, because the electrons technically kind of float somewhere within that electron cloud. But again, it's easier to visualize the electrons in the actual shells, so that's why we use the electron shell model for our purposes. Now here's a chart just showing you the charge, the location, and the atomic mass units, so how much the particles weigh or what their mass is. Now, protons, again, have a positive charge. Neutrons are neutral. Electrons have a negative charge. Now, an important differentiation here. Protons and neutrons weigh 1 atomic mass unit. Electrons do not really weigh anything. So the mass or the weight of electrons is kind of negligible. What that means is if you add electrons or take away electrons, you're not really affecting the mass or the weight at all, but you are affecting the charge. On the other hand, if you add neutrons or take away neutrons, you are affecting the atomic mass. So you are affecting that weight, but you're not affecting the charge at all. So neutrons and electrons are kind of opposite as far as adding or taking away goes. So neutrons do not affect the charge, but they affect the mass. Electrons affect the charge, but they do not affect the mass. And then again, protons and neutrons are in the nucleus. The electrons orbit around the nucleus in the shells. Now, the first shell has less energy than the outermost shells. And electrons, it basically takes more energy to keep them further away from the nucleus because they're trying to get towards that positive charge in the nucleus because opposites attract. So positive and negative attract. So it takes more energy to keep those electrons in their outermost shells. So that's why the outermost shells have more energy than the inner shell. And then again, that first shell is full with 2 electrons. The rest are full with 8 electrons. This is just showing you the periodic table of elements. We're going to talk more about this as we go through this. So, C, that is the atomic symbol for carbon. The 6 at the top is the number of protons, which is also equal to the atomic number. The 12 at the bottom is the atomic mass, which is the weight, so to speak, of the protons and the neutrons added together. So how do we determine the number of subatomic particles in an atom? Well, the atomic number is the number of protons. The atomic mass is the number of protons plus the number of neutrons. So we know the atomic number, that's the number of protons, so carbon would have 6. To get the number of neutrons, you just subtract the atomic mass, which is 12, minus the atomic number, which is 6. So that would be 6. Now, all of the elements in the periodic table have a neutral charge. So that automatically means the number of electrons equals the number of protons. So the number of electrons in carbon would also be 6. So when you're figuring out how many protons, electrons, and neutrons are in a particular atom, you need all of the information that you're provided in that picture up there on the left-hand side. So the atomic symbol again is C, which means carbon. The 6 at the top is the atomic number, which is the number of protons. And then the 12 is the atomic mass, which is the number of protons and neutrons together. So if you're given the number of protons, which you are, you can find out the number of neutrons by simply subtracting 12 from 6. So here's carbon, the picture. You've got 6 protons and 6 neutrons in the nucleus in the middle. You've got 2 electrons in the first shell, and then 4 electrons in the second shell, for a total of 6 electrons. So here's some other elements. You've got hydrogen, carbon, nitrogen, oxygen, sodium, and chlorine. So hydrogen, it's hydrogen, one on top, one on bottom. So hydrogen is the element. It tells you the atomic number is 1, which means there's 1 proton. It also tells you the atomic mass is 1. So if you want to find out the number of neutrons, remember the atomic mass is the number of protons plus the number of neutrons. Well, the number of protons is 1. So if the atomic mass is 1, 1 minus 1 is 0. That means it does not have any neutrons. However, we know it's a neutral element. So the number of protons is going to equal the number of electrons. So it's going to be 1, 1, and 0. Now, if you look at nitrogen, atomic number is 7. The atomic mass is 14. So you have 7 protons. You have the atomic mass is 14. So you have 14 minus 7 is 7. And then again, we know this is a neutral element. So 7 electrons as well. So 7, 7, 7. Now oxygen, atomic number is 8, atomic mass is 16. So the atomic number tells us the number of protons, which is 8. It's a neutral element, which means it has to have 8 electrons. And then the atomic mass is 16 minus 8, which means it has 8 neutrons. And I am just noticing that 14 and 7 are backwards. The 7 should be on top, the 14 should be on top, bottom, and the same thing with the oxygen. The 8 should be on top and the 16 should be on bottom. So I apologize for that. I will fix that for the future. So in a nutshell, the number of protons, electrons, and neutrons are not always going to be the same. Like these are nice and pretty, 777, 888, carbon was 666, but you can always figure it out the same way. So for sodium there, you're given the atomic number is 11, you're given the atomic mass is 23. So the atomic number is 11, which means there's 11 protons. We know it's a neutral element, which means there's 11 electrons. And then to figure out the number of neutrons, you just take 23 minus 11, which is 12. So you've got 11 protons, 11 electrons, and 12 neutrons for sodium. For chlorine, atomic number is 17, atomic mass is 35. So you've got 17 protons, which means you have 17 electrons, and then you subtract 35 or 17 from 35, and you get 18 neutrons. Now, atoms are neutral in the charge. Electrons equal to protons. That is the case for all of the periodic table elements. However, we do have something called ions. Ions are charged atoms. That means the number of electrons does not equal the number of protons. So for example, we have the sodium atom. The sodium atom has one little electron in its outermost shell at the top there. If it loses an electron, it becomes a sodium ion, and it gains that positive charge because it lost a negative charge. Now, if something loses a negative charge, it becomes more positive. If something gains a negative charge, it becomes more negative. Okay? So the sodium ion has 8 electrons in the outermost shell, and it's happy. So isotopes are atoms that have different number of neutrons. So carbon 12, carbon 13, and carbon 14. They have a different mass, so They have a different number of neutrons, but the electrons and the protons are equal. So they do not have anything messed with as far as the charge goes. Okay. So carbon-12 has 6 protons, 6 neutrons, and 6 electrons. But if you add a neutron, you get carbon-13, which has a different mass. You have 6 protons, 6 electrons, but you have 7 neutrons. If you add another neutron, you have carbon-14. Now you have 6 protons, 6 electrons, and 8 neutrons. So it has an even higher mass. Carbon-14 happens to be radioactive. It is not stable, but it can be used as a tracer in medical testing and can also be used to treat cancers with radiation therapy. So, ions have different charges. Isotopes have a different number of neutrons. So, the outer shell of an atom likes to be full. It can be filled in different ways. It can give up or donate electrons to another atom. It can accept electrons from another atom, or it can share electrons with another atom. Now, all 3 of these are going to end up in a chemical bond being formed. The electrons in the outer shell are the ones that actually participate in chemical reactions. So in a nutshell, the electrons in the outermost shell determine how reactive that particular atom is going to be. Atoms of inert chemicals have full outer shells and are non-reactive. And these are referred to in the periodic table as the noble gases. Neon is an example. If an atom has a full outermost shell, it can't react with anything because it can't accept electrons or do anything. So that's why they are called inert elements, meaning they're non-reactive. So here, this last column are the noble gases. They are all inert, which means that their outermost shell is full. So you got helium, neon, argon, krypton, xenon, radon, and that last one. Okay, molecules versus compounds. A molecule results when 2 or more atoms chemically combine. A compound is a molecule that contains 2 or more different elements. So molecules are the same atoms chemically combining. Compounds are different atoms chemically combining. Free radicals have unpaired electrons in their outer shell. Now, free radicals are very reactive and can actually damage the body. So superoxides. However, we can take antioxidants and inactivate these free radicals. So that's why you always hear about grapes, grape juice, wine, very high in antioxidants, so they can help neutralize these free radicals in our body. So we have oxygen, water, O2, and O2 negative. Are they molecules, compounds, or free radicals? Or elements? So oxygen, that's an element. Water. Now technically it's a compound, but water molecules are referred to often. O2. That's a molecule. 2 atoms of oxygen have chemically combined. And O2 negative. That's a superoxide. It has an unpaired electron in the outermost shell. Now, something important to note is not all molecules are compounds, but all compounds are molecules. So sometimes the terms compound and molecules are used interchangeably. Like water, for example. Water, H2O, it's a compound because 2 different atoms chemically combined, but it's also a molecule because atoms chemically combined. Okay, so chemical bonds. We just talked about the fact that we can fill outer shells and how we can do it, and in the process, chemical bonds are formed. So the first type of chemical bonds are ionic bonds. Ionic bonds are formed when atoms of elements are given up or accepted. So this is a type of bonding that's associated with inorganic molecules like sodium chloride. So if you look at the picture, you've got the sodium atom and the chlorine atom. The sodium atom has 1 electron in its outermost shell. The chlorine atom has 7 electrons in its outermost shell. So it makes most sense for sodium to donate that extra electron to chlorine, because then sodium will have a full outermost shell and so will chlorine. So sodium donates that electron to chlorine and we get the sodium ion and the chloride ion. Now the sodium ion gave up an electron, which means it gave up an electrical charge. So it became more positive. The chloride ion, opposite, it accepted an electron. So it accepted a negative charge. So it became more negative. So it's Cl-. So a cation is a positively charged ion, and an anion is a negatively charged ion. And when I was told cats are positive, and that's how I remember cations are positive. So sodium Na+, chloride Cl-, together you get sodium chloride. This ionic bond is going to form because there's an attraction between those positive and negative ions. Opposites attract. But just to show you how much it changes, sodium in its normal state is a silvery metal. Chlorine in its normal state is a noxious green gas. But you put them together and you get table salt and put it on your food. Electrolytes also form when an ionic compound breaks apart into its cations and anions in a solution. So the sodium ion, the chloride ion, that's the bicarbonate ion or the potassium ion. Now we have another type of bond, covalent bonds. Covalent bonds are formed when atoms of elements share electrons. Now this is associated with organic molecules. You can have single, double, or triple bonds. Single bonds, 1 pair of electrons is shared. Double bond, 2 pairs of electrons are shared. And a triple bond, 3 pairs of electrons are shared. Now nonpolar, the electrons are shared equally between atoms. So methane is an example. You've got a carbon atom and you've got 4 hydrogen atoms. Get CH4, it's a methane molecule. Everybody's sharing those electrons equally. Make sure you look at that picture of methane. You can see each hydrogen is sharing that pair of electrons with that carbon atom. Okay? Polar is an unequal sharing of atoms. This leads to electronegativity. Water is an example. So oxygen is a huge atom compared to the hydrogen atoms. So when the hydrogen atoms bind with that oxygen atom, the oxygen technically has a stronger pull on those electrons than the hydrogen atoms do. Because the oxygen has a stronger pull on the electrons, it becomes slightly more electronegative. So that's what the delta minus sign is next to the arrow you can see. It's saying it's slightly electronegative because it has a slightly stronger pull on those negatively charged electrons. Now the hydrogen atoms are slightly electropositive because they do not have as great a pull on that negative charge. Okay? Now keep in mind, all atoms want 8 electrons in their outermost shell to be full. So the carbon atom is one we're going to be talking about a lot because there's 4 electrons in its outermost shell, as you can see in that picture, which means it wants 4 more electrons. So it can make bonds with 4 other atoms. So it's a huge thing as far as living things are associated with. So covalent bonds, we got single, double, and triple. We got polar and nonpolar. So hydrogen atoms forming a hydrogen molecule. The structural formula is just H bonded with H. Molecular formula is H2. Then you've got oxygen atoms binding together. But oxygen, it has, if you count, 2, 4, 6 atoms in its, or 6 electrons in its outermost shell, which means it needs 2 more. So the oxygen atoms are going to share 2 pairs of electrons to fill their outermost shell. So that creates a double bond because there's 2 pairs of electrons being shared. So it's O double bond O. The molecular formula is O2. Now nitrogen, it has 5 electrons in its outermost shell, so it needs 3 more. So nitrogen atoms will actually share 3 pairs of electrons, creating that triple bond that you see in the structural formula. One line is a covalent bond, or 1 pair of shared electrons. So nitrogen is sharing 3 pairs of electrons to create that N2. Okay? And they're all nonpolar because they're all the same atom, so neither one has a stronger pull on either one. Now the last type of bond we're going to talk about is a hydrogen bond. Hydrogen bonds are very weak attractions found between molecules. Now that's an important note. It's between molecules. So it'll be between water molecules or within DNA. Hydrogen bonds are represented by a dotted line. So if you look at the water right there, you have a lot of H2O molecules binding together through hydrogen bonds. DNA, the 2 strands are held together with hydrogen bonds. But it's very important that those bonds are weak so they can be broken, because DNA needs to replicate itself. DNA needs to be transcribed, so it has to be unzipped relatively easily. In water, liquid water, those hydrogen bonds are constantly breaking and reforming, so the water molecules are floating around freely. So hydrogen bonds are between molecules, but covalent bonds are in the molecule. So I'll give you an example. Water, H2O. There are polar covalent bonds within the water molecule. So the oxygen and hydrogen are held together with polar covalent bonds. But hydrogen bonds are between water molecules. Okay, and you could be asked that on your test, so make sure you know. Hydrogen is between, polar covalent within. Okay? Okay, then we have chemical reactions. Now, chemical reactions basically are making and breaking bonds. Reactants are the inputs, products are the outputs. The arrow shows the direction of the product, so it shows the direction of what's happening. So here we have 2 hydrogen molecules plus an oxygen molecule, and it's going to result into water. Enzymes, remember, end in ASE. So any enzyme that's going to be involved will end in ASE for the most part. There are some enzymes that don't, but those come later on in the year. So the energy of activation is the energy needed to initiate that chemical reaction. Enzymes are catalysts because they lower the energy of activation. So basically, if you think about pushing a rock up a giant hill, an enzyme is going to come in and cut that hill in half. So now you don't have to push the rock up as high. So it's going to go a lot easier and a lot faster. And that's what catalysts do. Chemical reactions, again, are always happening in our cells, and this is called our metabolism. So this just shows you what I kind of just explained. The energy of activation, that's the energy needed to start the reaction. So an enzyme, as the one on the right-hand side shows you, the energy of activation without a catalyst is higher than with a catalyst. So an enzyme is going to come in, lower the activation energy required so that the reaction happens faster. That's just showing you no enzyme and with an enzyme. So we have 2 major types of chemical reactions: synthesis reactions and decomposition reactions. Synthesis reactions are building things. Decomposition reactions are breaking things down. So a synthesis reaction would be A plus B yields AB. An example is glucose plus glucose yields maltose. It is anabolic, which just means building up, and it is endergonic, meaning it requires energy. Decomposition is breaking something down. So AB yields A plus B, or maltose yields glucose plus glucose. It is catabolic, which means breaking it down. And it is exergonic, meaning it gives off energy. And if you think about it, creating something will take more energy than destroying something. Okay. So glucose plus oxygen yields water, carbon dioxide, and ATP. Is that synthesis or decomposition? Decomposition. You're taking glucose and oxygen and you're breaking it down to water, carbon dioxide, and ATP. You're getting energy out of the deal. Okay. So some concepts we need to know. When chemical bonds are broken, energy is released. So if you're breaking something down, you're giving off energy. If chemical bonds are formed, energy is required. So if you're building something up, you're going to need energy. The energy released during those decomposition reactions can be used to drive synthesis reactions. And this is what we call coupling. So if you have a decomposition reaction, AB yields A plus B, the energy that is given off from that reaction can be used to create CD. So you can use one energy output to drive the synthesis of something else. Now, metabolic reactions involve energy conversions because we need the cell to do work. Energy is just the capacity to do work, and it takes different forms. Potential versus kinetic. Potential is stored energy. So this is like the energy stored in the bonds of food, let's say. Kinetic is the energy of motion. This is mechanical and transport types of work. Now, if you think about it, if you have the potential to do something, you could possibly do it. So you have stored energy. But if it's kinetic, it's energy in motion. So potential energy is like water behind a dam. When you open that dam and release the energy, it turns into kinetic energy. So then we have the law of conservation, known as the first law, and the generation of heat, known as the second law. So we're talking about energy cannot be created nor destroyed, but we always lose energy as heat in the reactions. Now, inorganic molecules versus organic molecules. Inorganic molecules are associated with non-living matter, and organic molecules are associated with living matter. Inorganic are usually smaller than organic. Bonds in inorganic molecules are usually ionic. Organic are usually covalent. Now the exceptions there, water and carbon dioxide, are covalent bonds, but they are still inorganic molecules. What defines inorganic is it does not have a carbon bonded with a hydrogen. Organic has a carbon bonded with hydrogen. Okay, so if you're asked if something is organic or inorganic, you're going to look for carbon and hydrogen both together. Okay, so H2O, inorganic or organic? Inorganic. Glucose, organic. Carbon dioxide, inorganic. Sodium chloride, inorganic. Methane, organic. You have that carbon-hydrogen bonding. Now, both inorganic or inorganic compounds are vital to living cells, so that's important to note. Water is crucial to our survival, even though it's inorganic. Okay, so as I just said, water is vital for life. The volume of the cells and the body fluids comprise approximately 55 to 60% water. So most of our body is water. Water is a solvent. It dissolves solutes. So the solvent would be water. The solute could be NaCl. What you end up with in a solution is salt water. And this allows for transport of solutes in the body. So one property of water that is crucial to our survival is that it is a universal solvent, meaning it dissolves more substances than anything else on Earth. So things can be dissolved in water and then transported in our body. Hydrophobic, hydro means water, phobic means a phobia, like your fear. So hydrophobic means fearing water. Hydrophilic means loving water. So if something is hydrophobic, it is not going to dissolve in water. If something is hydrophilic, it will dissolve in water. So lipids, for example, are water-fearing substances. They are nonpolar molecules. Sodium chloride, on the other hand, is water-loving. It will dissolve easily in water. These are ionic or polar covalent molecules. Okay. Dehydration reactions, water is going to be an output. It is a synthesis reaction. In hydrolysis reactions, water is the input. It is a decomposition reaction. So if you think about it, if you are dehydrated, what are you lacking? You're lacking water. So when you take two monomers and take away water, you're going to combine them and make a bond, and water is going to be given off in the reaction. So a dehydration reaction is going to form a bond by removing water. Hydrolysis, on the other hand, hydro is water, lysis means to break. So you're using water to break a bond. So water is going to be the input, and it's a decomposition reaction because you're breaking a bond. So a dehydration reaction, you're creating a bond by removing water. Hydrolysis reaction, you're breaking a bond by adding water. And this just shows you have two monomers. You take out that water. Now you have a polymer and water as an output. You have a polymer, but you add water. Now you're left with monomers. So this is showing you dehydration, synthesis, and hydrolysis of carbohydrates and proteins, how the water is removed to form the bond, and then the water is added to break the bond. So arrow going one way, you've got dehydration synthesis. Goes the other way, you got hydrolysis. Sorry. Other properties of water. It has a high heat capacity and a high heat of vaporization. What this means is that it takes a long time for the temperature to change. So this allows for moderation of temperature. It also allows for organisms to survive in water because just because it gets really hot out doesn't mean the water is going to start to boil. Or just because it gets really cold out, the water is going to freeze. So it takes a long time for it to change temperature. Now, if you think about it, if you've ever put a pot of water boiling on the stove because you were going to make spaghetti or mac and cheese and you walked away from it, totally forgot about it. You go back like a half an hour later and there's still water in the pot. That's because it has such a high heat capacity and high heat of vaporization. It also serves as a lubricant. We have serous fluids and fluids in our joints that help us move around. So it serves as a lubricant to reduce friction. Okay, acids, bases, salts, pH, buffers. Inorganic compounds are classified as acids, bases, or salts. Inorganic compounds are going to ionize, or what's called dissociate, in water, and that releases ions, either cations or anions. So an acid is going to release hydrogen ions into the water, or H+. For example, HCl, like the first picture shows you. A base is going to release hydroxide ions, or OH negative, into the water, like KOH. A salt is going to release ions in the water that aren't hydroxide ions or hydrogen ions. For example, KCl. So if you look at the pictures, you have hydrogen chloride, hydrogen chloride dissociating into the hydrogen ions and the chloride ions. You have KOH dissociating into potassium ions and hydroxide ions. But then you have KCl dissociating into potassium ions and chloride ions. So an acid releases hydrogen ions, a base releases hydroxide ions, and a salt releases ions that are not hydrogen or hydroxide ions. A body has to maintain dynamic acid-base balance, and this is measured as the pH. The pH basically measures the concentration of hydrogen ions in a solution, and it ranges from 0 to 14. So the more hydrogen ions that are present in the solution, the lower the pH is. So if you look at the chart, a pH of 0 has the most hydrogen ions, and a pH of 14 has the least hydrogen ions. Now, hydrogen ions and hydroxide ions have an inverse relationship, which means that the more hydrogen ions there are, the less hydroxide ions there are. So something that has a low pH has more hydrogen ions and less hydroxide ions. Something that has a high pH has more hydroxide ions and less hydrogen ions. pH less than 7 is acidic, over 7 is alkaline or basic, 7 is neutral. So a pH of 7 is going to have an equal concentration of hydroxide ions and hydrogen ions. Less than 7, you have more hydroxide or more hydrogen ions. More than 7, you have less hydrogen ions. If you have less hydrogen, that means you have more hydroxide. If you have more hydrogen, that means you have less hydroxide. So an acidic solution with a pH below 7 has more hydrogen and less hydroxide. A basic solution with a pH above 7 has more hydroxide and less hydrogen. And a neutral solution with a pH of 7 has an equal concentration of hydrogen and hydroxide ions. Water is an example, either pure water or distilled water. So as the pH decreases, the concentration of hydrogen ions increases. Again, it's inversely proportional. A change in one pH unit to another yields an increase in hydrogen ions by a power of 10. So it's logarithmic. So a solution with a pH of 1 has 10 times more of the hydrogen than a solution with a pH of 2. And a solution of pH of 1 has 100 times more hydrogen than a solution of a pH of 3. So here is a table with some common substances and their pH values. So acidic, basic, or neutral? Blood. Blood is slightly basic, as you can see, 7.35 to 7.45. Gastric juice. As you can see, it is 1.2 to 3, which means it's acidic, highly acidic for the most part. Urine. 4.6 to 8.0, acidic to basic, and your urine concentration actually depends on what you eat, if you eat a lot of meat or not a lot of meat. Bile. 7.6 to 8.6 makes it basic. So, buffers help resist changes in pH and help maintain homeostasis. We have a few buffer systems in our bodies. The biggest one is the carbonic acid bicarbonate buffer system, and we will talk about that in the second semester. Last thing we need to talk about are organic compounds. We have carbon skeletons with functional groups attached. 4 categories: carbohydrates, proteins, lipids, and nucleic acids. You should have learned about these in biology, so hopefully you remember them. We have 2 general reactions that create them and break them down. Dehydration synthesis is going to make a large organic molecule from smaller ones. So you add monomers together to build a polymer. It is an anabolic chemical reaction. And remember, you're going to remove water in the process. Hydrolysis or decomposition reactions add water to break down large organic molecules into smaller ones. So you're going to add water to a polymer to break it down into its monomer components. And again, it's catabolic because you're breaking it down. So the monomers of the polymers, monosaccharides are the monomers for polysaccharides. So simple sugars combine to form complex carbohydrates. Fatty acids and glycerol molecules combine to form lipids, like triglycerides, fats, oils, steroids. Amino acids combine to form proteins, and we have a wide variety of proteins out there. And nucleotides combine to form nucleic acids, namely DNA and RNA. So, carbohydrates. The general formula is CH2O in parentheses with an N. It's usually a 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. Monosaccharides, glucose is used for that quick energy source. It is C6H12O6. It's a hexose because it has 6 carbons. Deoxyribose is the sugar base of DNA, C5H10O4. It is a pentose, and it is a pentose because it has 5 carbons. And then ribose is a sugar base of RNA. It is C5H10O5. That's a pentose as well because it has a 5-carbon base. Disaccharides are 2 monosaccharides together. Sucrose is glucose plus fructose, which is table sugar. Lactose is glucose plus galactose, which is milk sugar. And maltose is glucose plus glucose. This is just showing you the bases of the monosaccharides and the disaccharides. You have deoxyribose and ribose up top, then glucose, fructose, and galactose on the bottom. Disaccharides, you've got lactose and maltose. Now, isomers are something as well that you need to know. Isomers have the same chemical formula, but they're different structurally. A different structure means a different function. For example, glucose, fructose, and galactose all have the chemical formula C6H12O6, but they are all structurally different. If you look at the bottom of the first picture, you have glucose, fructose, and galactose, all different structures, but the same chemical formula. Then we also have lactose, maltose, and sucrose, all have the same chemical formula, C12H22O11, however, different structures. So isomers, same chemical formula, but different structures. And since they have different structures, they have different functions. Do not get it confused with isotopes, which have a different number of neutrons. 2 different things, okay? Then polysaccharides are a bunch of monosaccharides linked together. Glycogen, starch, and cellulose are all polysaccharides of glucose. Glucose is just linked together in different ways to form these different polysaccharides. So it goes to show you that even though they're all made up of glucose, if you link the glucose together in different structures, you have different compounds. So glycogen is used to temporarily store energy in the liver and skeletal muscles in animals. Starch is in plants, and it is used for an energy source. Cellulose is in plants, and it's used for their cell walls. For us, starch is used for an energy source sometimes, but it is very easily digestible. Cellulose is not digestible, so we use it for fiber in our diet. Then we have lipids. Lipids are hydrophobic, so they are insoluble in water, but they are soluble in alcohol. Fatty acids are the building blocks of triglycerides and phospholipids. Triglycerides are fats and oils. They have 3 fatty acid chains and a glycerol molecule. They store 2 times as much energy in the bonds as carbohydrates and proteins do, but we use them for long-term energy storage and insulation, as opposed to an immediate energy source. Adipose tissue stores triglycerides after you exceed your dietary intake of carbs, proteins, and fats. Now, we have 2 types of fat, saturated and unsaturated. Saturated fats are associated with animals. Unsaturated are associated with plants. Saturated fats are solid at room temperature. So just remember saturated, solid. Unsaturated fats are liquid at room temperature. Saturated fats are harder for the body to process, so we need to reduce our dietary intake. Unsaturated are actually better for our diet. Saturated fats do not have any double bonds between the carbons in their fatty acid chain. Unsaturated fats have double bonds in the fatty acid chain between carbons. So as you can see in the little picture, no double bonds in that carbon skeleton versus double bonds in the carbon skeleton. So here's a triglyceride molecule. You've got glycerol plus 3 fatty acid chains attached. Is that top one saturated or unsaturated? It's saturated. The middle one, that is saturated, and the bottom one, that is unsaturated. Now you can tell it's unsaturated because it has a double bond right here. When you are determining if something is saturated or unsaturated, do not pay attention to this double bond on the outside. That is there in every single fatty acid chain. You want to look for bonds throughout the tail here, in the tail. So here's a double bond. Double bonds will also kink the fatty acid. That's why this has a kink in it. Phospholipids are amphipathic. What that means is they have a hydrophobic portion and a hydrophilic portion. The hydrophobic portion is afraid of water. The hydrophilic portion loves water. So phospholipids make up our cell membrane here. You have the hydrophilic heads on the outside and the hydrophobic tails tucked away on the inside. So water loving on the inside and outside, water fearing tucked away and hidden in the middle. So this just shows you the polar head hydrophilic, the nonpolar tails hydrophobic. Steroids are also an example of lipids. They are 4 fused carbon rings. Examples are cholesterol, which is a small component of the plasma membrane, some hormones, estrogen, testosterone, cortisol, and some eicosanoids, which are short-distance hormones like leukotrienes and prostaglandins. We'll talk about eicosanoids more next semester when we get to immunity. And there is an example of the 4 fused carbon rings. So if you see that, you can always tell it's a steroid. Fat-soluble vitamins like A, D, E, and K, in excess, they can actually deposit in the tissues and start to become toxic to the body. So you want vitamins A, D, E, and K, but just enough for your body. You don't want to overdo it and bring in an excess, because then it can start to hurt you. Vitamin A helps your sight. D is for calcium absorption. You cannot absorb calcium without vitamin D. E is your skin and an antioxidant. K is for blood clotting. Proteins. Amino acids combine to form proteins. They are very diverse in function and diverse in structure. You have a chart over there that shows you the type of protein and the functions, and then some examples. They have 6 main functions: structure, regulatory, movement, defense, transport, and catalytic. Structural, for example, collagen and keratin make our hair and nails stronger. Regulatory, like hormones. Movement, like contractile, actin and myosin. They contract our muscles. Defense, so immunological, antibodies are actually proteins. Transport, hemoglobin transports oxygen. And then catalytic, like enzymes. As I said, the amino acids are the building blocks of proteins. So amino acids will combine to form dipeptides, which is just 2 amino acids together. They will then form polypeptides, which is multiple amino acids together, which will then form a protein. We have 20 different amino acids, and amino acids are actually named by their R group, so the remainder of the molecule chain. The basic structure of an amino acid is this. You have an amino group and a carboxyl group attached to a carbon, and then you have a hydrogen and an R group attached to that carbon as well. These are the amino acids, again, named by their R groups. They all have abbreviations as well as full names, obviously. You should be familiar with a few of these, like tyrosine, tryptophan, glycine, valine, methionine. We'll hear about those again. You don't have to know the structure or anything, though, don't worry. A peptide bond is the type of bond formed between amino acids during dehydration synthesis. So the water is removed and a peptide bond is formed. We have 4 levels of protein structure: primary, secondary, tertiary, and quaternary. The primary structure is just this sequence of amino acids. The secondary structure is the alpha helix or beta pleated sheets. These form because hydrogen bonds start to interact. Then we have the tertiary structure, which is actually the 3D globular shape. This is formed because R groups start to interact. And then you have the quaternary structure, which is more than one polypeptide chain together. Hemoglobin is an example. Hemoglobin has 4 polypeptides, 2 alpha chains and 2 beta chains. Proteins, enzymes of course, must maintain a 3-dimensional shape in order to be functional. So if they lose this shape, they are no longer functional. And as I said, hemoglobin is an example with alpha and beta chains. We have 2 basic types of proteins, fibrous and globular. Fibrous are insoluble in water, but they are structural in function, like collagen and keratin. Globular are more soluble in water but are metabolic in function, antibodies, hormones, enzymes, etc. Now, denaturation refers to a protein losing its 3D structure and no longer becoming functional. So if you have a normal protein like this, and then something happens and it unfolds to this, now this protein is no longer functional. The loss can be permanent or it can be reversed depending on what caused the denaturation in the first place. Some things that might alter the structure of a protein: temperature, pH, and ion concentration. If you go from tertiary structure back down to secondary structure, now this protein isn't going to be able to function anymore. Changes in temperature can denature a protein. If the temperature gets too high, especially, that will denature a protein. That's why it's super important if you have a really high fever to get to the hospital, because they have to bring it back down or your proteins can be denatured permanently. pH. Most enzymes have a narrow pH range that they can only function within. The whole point of enzymes is to speed up chemical reactions. So the basic enzyme reaction is the enzyme plus the substrate is going to yield the enzyme substrate complex, which will then yield the enzyme and the product. Enzymes are only needed in small quantities because they are reused. Now notice in this one, sucrose and water are the substrates. The enzyme is sucrase, ASE, it ends in ASE. The products are going to be glucose and fructose because we're breaking down sucrose. And then the enzyme is going to go on, and this will continue on until the enzyme is no longer needed. So we only need to make enzymes in small amounts because they can be reused, which is a good thing. Enzymes are very specific. So enzyme, the active site to the substrate, again, they end in ASE. And the induced fit model just states that the enzyme, the active site, will kind of fold around to cuddle in the substrate. They're very efficient, but they are subject to control. Through feedback inhibition, once we have enough product made, the product can then go back and bind to this enzyme to stop the reaction from happening. So what do we have here? Is this dehydration or hydrolysis reaction? It is hydrolysis because we are using water to break down sucrose. Synthesis or decomposition? Decomposition because, again, we're breaking down sucrose. Okay, the last thing is nucleic acids. The basic structure of the nucleotide, which is the monomer for a nucleic acid, is a sugar, a base, and a phosphate group. The sugar can be deoxyribose or ribose, and the base can be cytosine, adenine, guanine, thymine in DNA, or if we're talking about RNA, instead of thymine, it's uracil. Now, purines are adenine and guanine, pyrimidines are cytosine, thymine, and uracil. The way I remember the difference is pyrimids can cut you. Cut cytosine, thymine, uracil. Pyrimids can cut you, they're pyrimidines. Now this is just showing you the difference between DNA and RNA. The sugar is deoxyribose in DNA, ribose in RNA. The bases, adenine, guanine, and cytosine are in both, but thymine is in DNA, uracil is in RNA. There's one phosphate group in both. DNA, however, is double-stranded, attached by hydrogen bonds. RNA is single-stranded, but they both have that sugar-phosphate backbone. So you got sugar-phosphate, sugar-phosphate. Function, DNA stores genetic information to make proteins. RNA actually carries out protein synthesis. ATP, adenosine triphosphate, is a nucleotide, not a nucleic acid, but the function, it carries out just enough energy for the cell to be able to do work. So all of the chemical reactions that happen in the cell, many of them require ATP or ATP is produced. So here we have adenosine triphosphate. It is adenosine, ribose, and 3 phosphate groups. This last phosphate group is not very stable and it has a lot of energy in it. So it falls off readily. So when it falls off, you're left with ADP or adenosine diphosphate, plus a phosphate group, plus energy. And we'll talk more about what happens to this phosphate group later on in the semester. It's a reversible action, and it's actually called the ATP-ADP cycle. So ATP broken down to ADP and phosphate by ATPase. It is an exergonic reaction, so energy is released. ADP needs to be recharged, so you add the phosphate group and energy to create ATP. ATP synthase is the enzyme that does this. So ATP synthase synthesizes ATP. ATPase breaks down ATP. Okay? And that's endergonic, meaning it requires energy. ATP is generated through cellular respiration. The anaerobic phase does not require oxygen, but you only get 2 ATP molecules from 1 glucose molecule during glycolysis, so it isn't very efficient. The aerobic process requires oxygen, but you can generate up to 34 ATP molecules from that 1 glucose molecule during the Krebs cycle and the electron transport chain. So glucose plus oxygen yields water, carbon dioxide, and ATP. The Krebs cycle yields about 2 ATP, so it's really the electron transport system that gives you the most ATP.